So now we actually know a bit about how mitotic spindles form, but there's another step that is quite important in the formation of mitotic spindle, that is the dissolution of the nuclear membrane. Now, why does this happen? As you look at the structure right here, you can notice that the chromosome is usually present. Sorry, the chromosome is. The chromosome is usually found inside inside the nucleus, of course, and the centrosome is responsible for the formation of mitotic spindles in order to pull the chromosome to wherever it is needed to be pulled to, is found inside the cytoplasm. So it's important during, especially during late prophase, to sort of merge to get rid of the boundary that is found between the chromosome and the centrosome in order to facilitate the movement of mitotic spindle from the cytoplasm into the nucleus. And this entire process is known as the dissolution of the nuclear membrane and the nucleus. Dissolution is brought about by the removal of nuclear pore, and then the nuclear lamina, and lastly the nuclear membrane. And this is all carried out by a checkpoint, by surpassing a certain checkpoint, and the activation enzyme, also known as the BCDK1. So BCDK1 induces the degradation of the components of the nucleus. So after this takes place, the nucleus looks somewhat like this. So as you can see that the entire membrane is gone, and then you can see there are like certain small remnants of the membrane. Then these are also known as vesicles. Essentially, the remnants of the nucleus and the membrane are packed into small vesicles, and we really don't know what actually happens with these vesicles. There are some speculations that maybe they are degraded by the by the endoplasmic reticulum, or it can no, it's just one speculation. There's a speculation that is degraded by the endoplasmic reticulum. Now, it's quite surprising to note that the endoplasmic reticulum is the only structure that persists throughout the mitosis. Throughout mitosis, we have another structures like mitochondria and Golgi bodies that they usually form copies of themselves so that they can divide and then incorporate themselves into each of the daughter cells equally. So let us take a look at a phase of the cell cycle, especially in the M phase, known as pro-metaphase. Now we know for the fact that in metaphase there is an alignment of the chromosome at the equatorial region of the cell. So this alignment does not really happen naturally. Because chromosomes are not naturally present on the equator. They are scattered inside the cell. They might be here, anywhere inside the chromosome. So they are inside the cell. So the chromosomes are bound together by a certain entity known as the centriole. So this is a centriole, and centriole is present inside a structure known as the centrosome. And the work of the centriole is to make mitotic spindle in the form of microtubules. So this is how there's a progression of mitotic spindle outside. But now why do this mitotic spindles, why do they originate from the centriole? What is their actual job? So their job is to search for chromosomes so that they can pull the chromosomes towards themselves to wherever place that they are necessary, that they are to be in the particular phase that is concerned. So in this phase, pro-metaphase, there's a need for the chromosome to be aligned at the equator somewhere here. So it keeps on searching for the chromosome inside the cell. So for example, the chromosome is located here. Now, once there is a contact of the mitotic spindle, now whenever we talk about the contact of the mitotic spindle with the chromosome, we essentially see that the mitotic spindle might come from here and attach to the chromosome right here, and then pull it, in a pretty straightforward manner toward itself. But that's actually not the case. So just imagine that this entire thing goes and searches for the chromosome. But when it finds the chromosome, it attaches itself in a parallel manner. So, this entire structure is sort of sideways, as you can see there. And after after the attachment, to this mitotic spindle, this entire chromosome, it sort of slides down the length of the mitotic spindle. But however, it does not really stay that way. It brings itself back into position and sort of attaches itself at at the plus end of the mitotic spindle. So this is the minus end and the plus end, and then there is a proper attachment of the chromosome right there at the kinetochores. So after this, I would also like to add that there's just not one pericentriolar body. There's not just one centriole. There are two centrioles that are present on the opposite ends of the cell. So this is the cell. There's a centriole here, and then there's a centriole and a centrosome here, and they search for the chromosome inside the cell from both the ends. So when one end of the chromosome is attached, one end of the kinetochore is attached to a chromosome essentially has two kinetochores. So one end is attached here, and then there's the other end which will be attached by another centriole. And then the means by which it will be attached is by the spindles that project, the mitotic spindles that project out of the chromosome, and then they find the kinetochores. So as you can see, if there are two strings pulling a certain entity towards themselves, there's going to be a tension, and then there's going to be a movement of the cell, of the chromosome towards either of them, and this will render the entire chromosome to sort of associate itself into the midline. And then there's another phenomena that is noted in the case of the attachment of this mitotic spindle on the chromosome. At a certain point, a certain mitotic spindle can give rise to a lot of mitotic spindles. So if there's a detection of a chromosome and a stabilization after the attachment to the kinetochores, there are certain other spindles that originate from the primary spindles and attach themselves to the kinetochores. And hence, after that, there is, a neat arrangement on the equatorial plane of the chromosome with the help of the two centrioles. And this is also known as congregation of the chromosome.